So now we're recording. If you're watching the video, you can go ahead and probably skip about 4 minutes in, because we are 4 minutes before the lecture. Mathis is here. And then Rain's in the back, right? Good morning. I'm sorry.
Yeah.
And Dow. Well, we've got less than a minute left, so I'm going to do the reminders because there's really nothing new here. Just me reiterating stuff. So, again, pre-labs are past due now. As you've noticed, I'm getting stricter and stricter. So, if for some reason you haven't done all 3 parts of the pre-lab, do it now. Because when I'm done here, I'm going to go and grade them. 3 parts. I mean, the video questions, the regular questions, and then your hypotheses. And remember, the hypotheses are just multiple choice, so all you have to do is bold them or delete the other options. I think it's one of the easier pre-labs you've had so far, despite the fact that there's 3 parts to it. So yeah, there's that. The exam is due on Sunday. It covers chapter 1 and 2. I mean, obviously you can do it now if you want. I think it should be open, but we're still covering chapter 2, so I would recommend it that you can if you want. It's going to be due online like you do with the quizzes. It's 50 minutes to take 50 questions because it's a closed book exam. So in theory, you wouldn't have time to look up those answers. And, again, 1 minute per question. But that being said, if you want more time to take it, like if you're not a good test taker or you're nervous or whatever, let me know. I can meet you online. As long as I can see you and hear you and see your screen and know that you're taking a closed book exam, I don't care how long it takes you to take. You can take all day. That doesn't matter to me. I'll be there for you. I'm not going to sit there and stare at you the whole time. It would be as if you were taking an exam in the room where the instructor doesn't just stare at you the whole time, but they're walking around and look occasionally to see what you're doing. So same situation there, so let me know. Independent work is now down to 15 points per page. So, get to writing. You're running out of, I don't say running out of time, you've got technically all semester, but Obviously, the value is going down as time goes on. And then it reminds you, let me help with anything you need help with, right? If we're talking about a pre-lab or a lab or a quiz, let me help you, right? So if you're doing something, you don't have to do it alone. I'll meet you online. I can help you go through it. Or before you turn something in, spend 5 minutes, meet me online, let me look it over, and be like, oh, ooh, man, you missed that. Make sure you get that. Or anything that you've already done, you can meet me and let me give you feedback, right? If you let me give you feedback, not only will that help you because you'll learn, but also you get a 5% boost to the grade. So whether it be a quiz that you've already done or a lab that you've already done, let me give you feedback. 5% boost to the grade. That's it. Anything else? Do you guys have anything? The announcements, reminders? All right. Well, let's get started. In theory, we'll get started. There we go. All right. Here we go. So, again, we're talking about biological molecules. We talked about, well, before we get started, first word for minutes is going to be avocado. Since that's what's on the screen right now, avocado. Anyway, we're talking about biological molecules. There's 4 main categories. The first one we talked about was carbohydrates. We're done talking about that. Now we're talking about lipids. Lipids are different from carbs in a lot of ways, and they're different than proteins and nucleic acids. So I don't know if you remember, but I was telling you when we first started this conversation last week, I was saying, technically, most of what we're going to talk about are polymers, right? There's polymers that are made up of repeating monomers. And I said that there was one exception, and this is the exception. So lipids are not technically polymers. They are made up of different parts, but the different parts are not technically monomers because they're not the same repeating parts. They're actually different parts. They're also not necessarily huge compared to some of the other stuff we talked about. But let's talk about it. You don't necessarily need to know any of that for the exam. This is kind of like the introduction. There's different types of lipids. The first one you need to know about is fats. Are fats. You need to know a triglyceride is what we call fat. Again, like I was saying, technically it's not a polymer made up of repeating monomers, but it does have parts, right? There are parts that it's made of, and you need to know the parts. So the parts are a glycerol molecule and 3 fatty acid molecules. And I'll show you a picture of it on the next slide. But yeah, that's what a typical fat or triglyceride is made of. You need to know that. The fatty acids are very rich in energy. As a matter of fact, last week when we started this conversation, I showed you a picture of octane, gasoline that we burn for fuel and energy for our vehicles. And then I showed you a picture of a fat. So here we are, we're back to that, right? So we're talking about a fat. There is 2 times the energy in a fat molecule than there is compared to a carbohydrate. Of course, that's pretty vague, right? Because it depends on which specific type of fat and which specific type of carbohydrate, but that's in general. You can look that up for independent work if you want to get more specific. I put it next to that because that's nothing that you need to know for the exam. The point is, generally speaking, fats are they have more energy than carbohydrates. And fats are stored in adipose cells. Does anybody know what adipose cells are? Not that I expect you to. Just curious if anybody knows. I'm also going to put it next to that because you don't need to know that either. But fat. So when you think of fat, like that right there, that's also fat, right? Adipose cells. Too busy grading. I don't have time to take care of myself anymore. Anyway, any questions so far? Next slide, I'm going to show you a picture of a triglyceride. And that's what it looks like before it comes together. If you download the PowerPoint, you can watch that. You can see an animation of those things coming together. Because right now, what we have is 4 different molecules, right? 1, 2, 3, 4. Technically, that's not a triglyceride yet until they all come together. So, you need to know this for the exam that's due on Sunday. What kind of reaction would it be when all these things come together? When you're building something and not breaking something down. In this course, what kind of reaction do we call that? Here's a hint. Now you can see it. There's the reaction happening. What kind of reaction is that? Last time I asked you guys, I said you didn't. I didn't expect you to know it. I kind of do expect you to know it now because you're about to have an exam on it. Whenever you build molecules in this course, when we talk about chemistry, when you're building a molecule, bringing things together, it's dehydration. When you break something down, that's hydrolysis. So yes, that's a dehydration reaction. And this was the hint, because in the reaction, once you have water in the products, you've dehydrated, so to speak, the reactants. Therefore, it's a dehydration reaction. Well, at least that's the way I like to think of it to help me remember. But anyway, yeah, dehydration reaction. So, any idea why our bodies need fat? Think about it. Think about the fat that's on you, a lot on me. What does your body need it for? Like, what's it for?
It's like reserve or something.
Reserve is good but a reserve for what?
Energy.
Good to store energy. What else? There's two other big functions.
Isn't it good for your brain?
Oh, actually, that is true too, but that's not one of the big three that I'm going to mention. But yeah, your brain is mostly fat. So yes. But no, that's not one of the other two that I was talking about. I give you an inappropriate hint. More cushion for the pushing is the biological term. Well, yeah, basically. Energy storage, like you already said, cushioning, like you said, for the butt. Also, and people don't think about this, but the bottom of your feet also have fat for cushioning. And also insulation. And then, I think humans don't think about the insulation part a lot because, we have heaters and clothes, so we have other ways of keeping ourselves warm. But think about like walruses and polar bears and stuff, right? That's why they have fat, to keep them warm. Anyway, any questions about the functions of fat? All right. How many of you have heard the term saturated fat or unsaturated fat? You guys know that, right? Well, there's a difference between the two, and you're going to need to know the two. So unsaturated fat, it's called unsaturated because it doesn't have as many hydrogen atoms as it could. So when we're talking about saturation, when something's saturated in this context, that means it's saturated with hydrogen. And I'm sure you've heard the term saturated before, even outside of this context, right? Like if somebody were, working in the yard, they get really sweaty and their shirt's just completely soaked, you might say their shirt is saturated with sweat, meaning there's just so much sweat they couldn't absorb anymore, right? So saturated means like basically you just can't absorb anymore, so to speak. That's the same situation here. But in this case, we're not talking about sweat, we're talking about hydrogens. Unsaturated fats are often called oils, so I'm definitely not going to ask you that. I might ask you about this part, that they're liquid at room temperature. It's important, it's definitely important. So we'll talk about that for a second, but I'm not sure if I'm going to ask you that. Liquid at room temperature. The reason being, look, you can see this. Here's an unsaturated fat. Down here, here's a saturated fat. Now, first of all, not all unsaturated fats look exactly like that. They all have at least one double bond, because that's what makes them unsaturated, because they don't have all the hydrogens that they could, right? So they have at least one of those, but the area that they have it is always different, right? So it's not always that specific, those two specific carbons that have the hydrogen bond. Also, sometimes if there's two carbons, right, it could be more than one double bond. My point being, imagine trying to stack a bunch of these together and a bunch of those together. Which one do you think would be easier to just stack on top of each other? Saturated or unsaturated? Saturated. Good, right? And that is actually how it is. And I mentioned this before, and I know we're not chemists, but you can look at a molecule and common sense can actually tell you a lot about it. And in this case, that can tell you something, or common sense can tell you a lot about that saturated fat. Those stick closely together. It's nice and easy. They just fit nicely together because it's nice and straight. But because of that, they're often solid at room temperature. Saturated fats are solid at room temperature. And because of that, that's why they're unhealthy. Those are the fats that clog up your arteries because they do such a good job of just like clogging you together, right? Just stacking up on top of each other. Unsaturated fats are the opposite, right? So they don't fit together nicely. So because of that, they are usually liquid at room temperature, which isn't that important. But what's important about that fact is that because they're liquid at room temperature, that again, they're just harder to stick together Therefore, they're healthier, right? Because they don't clog your arteries, because they don't stick together as nicely as the saturated fats do. Yeah, so that's it. And then the other, sorry, really quickly, there's also polyunsaturated fats, which is what I was getting at earlier. So here you can see there's one double bond, right? But sometimes saturated fats have more than one double bond, and that would be called a polyunsaturated. Also, keep in mind, we're talking about triglycerides. It's a horrible drawing, but remember, so you've got your glycerol molecule and then your 3 fatty acids, right? All you need is for one of those fatty acids to look like this, and then technically the whole thing is considered an unsaturated fat. Meanwhile, to be considered saturated, they all have to look like this. They all have to be saturated with hydrogens, no double bonds. Yeah. So any questions about that? All right, here's a picture. This is not from your textbook. This is from the other textbook. It's a good picture. It just goes to show how, again, fat can clog your arteries. Generally speaking, saturated fats are unhealthy. But anyway, and also, generally speaking, it's usually animals where you're going to get the saturated fat. So when you're eating bacon or pork, even a little bit of chicken, right? When you're eating meat, basically that's where you're getting your saturated fat. Generally speaking, when you eat other stuff, you don't get saturated fat. Anyway, any questions about that? All right. So like I said, generally speaking, when it comes to your diet, meat is where you get saturated fat. So on the other side of the coin, it's usually plants and fish that give you the unsaturated fat, which I know fish is an animal, so it's kind of stupid the way I just put that up there. But yeah, fish and plants is where you generally get unsaturated fat. Again, like I said earlier, they're liquid at room temperature, which sounds trivial, but again, that indicates that the molecules don't stick together very well, which means they're generally healthier. And tropical plants are the exception to that rule. So again, most plants are high in unsaturated fats, but for some reason, tropical plants are the exception. So you will get things like coconut oil, for example, is high in saturated fat. Anyway, any questions about that? All right, here's another picture that's not from your textbook. I'll put a big old X through this. You do not need to know this for the exam. But yeah, that just kind of shows you the difference, for example, between corn oil and palm oil and butter, which is another good independent work topic, right? So what about olive oil? What about Crisco? There's so many other things that are not on there that you could look up, see what they're made of. How much is unsaturated, how much is saturated, how much is monounsaturated, as in like there's only the one fatty acid that's unsaturated. Polyunsaturated, again, I mean, there's more than one of those 3 fatty acids that are unsaturated. You can look at all that at friend and work if you want. You don't need to know it for the exam, but there you go. Any questions about that picture? All right, let's talk about trans fats. So again, a saturated fat is saturated in hydrogen, right? It's carbons that have all the hydrogens they can because they don't have any double bonds between them. So it's a bunch of hydrogens that have saturated it. But manufacturers can convert unsaturated fats to saturated fats. And they do this in a process called hydrogenation, which makes sense, right? Because they're adding hydrogen atoms to it. Why do they do it? They do it because they don't want the food to spoil as quickly. It's also very good for frying, and it can make those things solid at room temperature. So, for example, margarine, right? So margarine is basically an oil that they hydrogenated, and therefore it can become solid at room temperature. And this can create something called trans fats, which are very bad for your health. If anything, as far as the exam is concerned, that's the bullet point you need to know. And that's it. You just need to know that trans fats are bad for your health. I'm not going to ask you about hydrogenation. I'm also not going to get specific about why trans fats are bad for you on the exam. But you can look it up if you want for independent work. Because look, I'm barely scratching the surface, and I'm sure you guys have heard of trans fats before. Why are they so bad? I told you a little bit why they're bad for you. It increases the bad cholesterol. But why else is it bad for you? What makes it worse than other stuff? Look it up for independent work if you want, but I'm not gonna ask you about it on the exam. Any questions about that? All right. So then let's talk about omega-3 fatty acids. I'm sure you've heard of those. Those are really good. These are the good fats. They are required, right? You need them in your diet, but we cannot make them. Like your body cannot make omega-3 fatty acids. So you have to get them through your diet. Yeah, they reduce heart diseases, they reduce arthritis symptoms, they reduce irritable bowel syndrome, and usually they're found in oily fish and nuts. Like walnuts are really good for it, for example. Salmon, really good for it. But again, we're just scratching the surface. So for independent work, you can look deeper into that. Exactly why are omega-3 fatty acids good for you? I gave you a few things that they do, but how? How do they do those things? You can look that up for independent work. And which particular fish, I mean, I gave you two examples, but which particular fish are really high in fatty acids? Which particular nuts are really high in fatty acids? These are all things that you can look up for independent work, which again is now down to 15 points per page. So, now's the time to do it. Any questions about that? All right. Now we're moving on to a different type of fat. So we're done talking about triglycerides. Now we're talking about, let me reiterate. Everything we've talked about previously today, those were all under the category of triglycerides, right? Those are all those types of fats. Now we're talking about something called phospholipids. These are the things that make up your cell membrane. So instead of having 3 fatty acids, these things have 2 fatty acids. So you've got your glycerol molecule, like we did before, 2 fatty acids, but now that's also attached to a phosphate. And you might need to know that, but again, I'll remind you, these are multiple choice questions. So it's not like there's going to be a question where I say to you, like, Carla, tell me the things that make up a phospholipid, right? It won't be like that. It'll be more like, hey, there's this thing. It has 2 fatty acids and a glycerol attached to a phosphate. What kind of molecule is it? And you know, yeah, that's a phospholipid. The interesting thing about these things is they're both hydrophobic and hydrophilic, meaning they repel water. Some parts of the molecule repel water. Other parts are attracted to water. That will be important later in the semester. I think next chapter, as a matter of fact, we're going to finally get out of talking about chemistry and stuff like that. And we're actually going to talk about how cells work. And one of the things we're going to talk about, obviously, is the cell membrane. That's what makes the cell. So the fact that it's both hydrophobic and hydrophilic will be important when we have that conversation. Anyway, any questions about phospholipids? Okay, so this slide is actually out of order. It should have been after this next slide I'm going to show you, because these are the fats that we finished talking about. So now we're going back to talking about triglycerides, right? So again, triglycerides, you have saturated and unsaturated. Generally speaking, saturated is bad for you. Generally speaking, unsaturated is good for you. Generally speaking, there are exceptions. For example, you can look up the difference, or look up, if you want, for independent work, margarine. How is it bad for you? You can look that up. Obviously, donuts, right, are not good for you. So again, I'm saying, generally speaking, saturated is bad and unsaturated is good, but there are exceptions. Also, what is it? Man, I'm trying to remember some stupid thing I saw on social media. So remember, like I showed you that picture of, when we started this conversation, I showed you a picture of octane, which is gasoline, and I showed you a picture of fatty acids because they look very similar. But I've seen some stupid meme about, like, the stuff you're eating is only one atom away or one molecule away from being, I don't know, paint or something stupid, right? But again, I want to remind you, like, that's how chemistry works. Yes, you can only be one molecule away from something, but it is one molecule away from something, so it could be completely safe. Anyway, any questions about that picture? All right. So the next type of fat, so again, we've talked about triglycerides, which are here. We talked about phospholipids, which should have came after this slide. Now there's a third type we're going to talk about. They are steroids and waxes. Steroids. Actually, I don't know. Probably should have put that at the beginning of the slide too. So far, everything we've talked about, other than the phospholipids, are hydrophobic, right? For the most part, all fats are hydrophobic, meaning they repel water, right? That's why when you mix oil and vinegar, they don't actually mix, right? Because vinegar is water-based, oil is fat, so they don't mix. But yeah, steroids are hydrophobic. They are different than the other fats that we've talked about, both in their structure and their function. Here's something that you'll need to know for the exam. Speaking of their structure and their function, they're composed of a carbon skeleton that has 4 fused rings. You need to know that. And again, it's a multiple choice question in a 100 level course. So I'm not going to say, Carla, tell me how many rings are there, right? It's not going to be like that. It's more like, again, the question's like, I'm going to describe something, and based on that description, you need to recognize it as a steroid in this case. Speaking of steroids, the next word for attendance is baseball. Sorry, for me, when I think of, I know there's a lot of athletes that do steroids, but for me, due to my old age, when I think of steroids being abused, I think of baseball. Mark McGwire, Sammy Sosa, that kind of
One
stuff.
of them like Cincinnati Reds too, or something.
Yeah. What was his name? Barry Bonds, maybe?
Yeah, it's like the Redskins, no, what is it, the Cincinnati Reds,
Mm-hmm.
one of the big players, he was a big guy.
Yep.
Bill, got hot stuff.
But anyway, yeah, sorry, I got off on a tangent. So anyway, yeah. I'm going to show you a picture of a steroid, so you'll see that carbon skeleton, so you'll understand what I'm talking about. But yeah, that's one thing you need to know. They vary in their functioning groups, right? So steroids, they all have that 4-ring structure, but they're different based off of what's off of that structure, right? So they have these different functional groups that we talked about last week. So all these different things that are sticking off of them. And that actually makes a big difference. Because think about like the different, and we'll have this in the slide that's coming up, but think about the difference between testosterone and estrogen, right? One of them makes you a male, the other one makes you a female, or I should say, facilitates that. But they're both steroids, right? And they're only slightly different. They're definitely the same as far as this part. They all have the same 4-ring carbon skeleton. But what's different is those functional groups that come off of them. Here's what it looks like. There's the steroid skeleton. 4 rings. So again, all steroids have this basic shape, but they have these things, different things that stick off of them. And the things that stick off of them is basically how they're defined. Again, using the same example, that's what makes testosterone different than estrogen. It's the things that are sticking off of it. Anyway, any questions about steroids? All right. Next thing we're going to talk about is another type of fat. It's called cholesterol. This is another key component of cell membranes, just like the phospholipids that we talked about earlier. Cholesterols are a key component of your cell membranes. And cholesterol is the base steroid. So all the other ones, like the testosterone and the estrogen I was talking about previously, they're all based on cholesterol. So we take that cholesterol and we make some adjustments to it, and that's where we get the other steroids, which is another good independent work topic. Again, I'm not going to go that deep on the exam, but you could ask yourself, how? What's the name of that chemical reaction? Where we go from cholesterol to testosterone, for example. Where does that happen? Which enzymes? You haven't learned about enzymes yet, but you will soon. Which enzymes are responsible for those chemical reactions? Anyway.
And the enzymes is what breaks down.
Good question. We're going to get there. Enzymes are basically, and you'll need to know more later, but for now I'll just say, enzymes are the things that make the chemical reactions in your cells happen. Without the enzymes, for the most part, they won't happen. They're they're very specific. So any chemical reaction I talk about, there's a specific enzyme that does that chemical reaction. And yeah, we'll get to that. Anyway, any questions so far about cholesterol? Again, like I said, that's the base steroid, right? So here's cholesterol right here. And then you can see, depending on what you do to it, it can become testosterone or it becomes estrogen. And look at the differences between testosterone and estrogen. Right here, we have that oxygen with a double bond. Here, instead, we have an oxygen and a hydrogen. Here, we have an oxygen and a hydrogen. Here instead, we have an oxygen with a double bond, and that's it. So those differences are what makes largely the difference between a male and female. Just those small differences in that molecule. Which is crazy when you think about it too, because when you really think about it, they would have the same chemical formula, right? Because they both have two oxygens, what I'm referring to, two oxygens and a hydrogen, right? Two oxygens and a hydrogen. It's just a matter of where are those two oxygens and hydrogens. And that small difference makes the difference between, generally speaking, males and females. Anyway, any questions about that? All right, this is from the old textbook, so I included it. I just kept it with PowerPoints. I'm not going to test you on it, but like I was talking about earlier, for me, because I'm so old, when I think of steroids, I think of baseball. But yeah, people do abuse steroids. There are synthetic anabolic steroids. They're not all bad. Sometimes you need them. I guess I can't ask because of HIPAA, but I can tell you I've been prescribed steroids. Those for poison ivy. So like when you I'll get to you in a second, when you're like, when you need to build up muscle for some reason, maybe because you have poison ivy or because you have cancer, whatever it may be, you can be prescribed steroids. Yeah, anyway, question?
I was gonna ask, ain't inhaler steroids?
Which one?
An inhaler?
Oh, yes, there are inhaler steroids, yeah. That's a good point. You can look that up for the work too. Not that you need to know that, but yeah, they have like, what is it? I used to be a pharmacy tech, and my daughters have asthma. I should know this. Man. Anyway, albuterol. You know what's albuterol?
Yeah.
The albuterol is an inhaler. I'm just my daughter's on multiple inhalers. I don't remember which one's which, but yes, there are also inhalers that have steroids. Yes. Anyway, yeah. So steroids are good. They can be bad. Yes, ma'am.
So when you're talking about steroids, the bad and the good, is there a difference in the chemical breakdown, or is it just a steroid that is given to you
Good question.
in the right
Yeah, yeah, exactly. Yeah. So there's no difference in the molecules. It's the same thing. It's the same drug. It's just how you use it. Whether you use it or abuse it, that's what makes it good or bad. Good question. Any other questions? And of course, also, I want to reiterate really quickly. This is from the old textbook. I just kept it because I already had it on the PowerPoints anyway. Not in the textbook. I'm not going to ask you anything about it. Same thing with this slide. Just some more information about synthetic. But no, that was it. Yeah. So anyway, yeah. I'm not going to ask you any questions about that. Any more questions about steroids? All right. Let's talk about yet another category of lipids. Wax. Wax. These are hydrocarbon chains that have an alcohol group, or OH. So, and I've talked about that previously too. I pointed out some OH things before. Now you know that's an alcohol group. So hydrocarbon with an OH group and a fatty acid. That's what a wax is. And I can almost guarantee you I'm not going to ask any questions about that, but just so you know, waxes, when it comes to animals, we get it from beeswax and lanolin. When it comes to plants, they use it to protect their leaves. That's it. And again, I'm not going to ask you any questions about waxes. So much stuff. There's more important stuff in the lipid category that you need to know about. Triglycerides, phospholipids, steroids, stuff like that. Wax, I'm not really concerned with. But there it is. Any questions about wax? All right, let's move forward. This one, in my opinion, is one of the more important ones. This is tied for the most important of the categories of carbohydrates I'm going to talk about. So we talked about carbs, we're done with that. We just finished talking about lipids. Now we still need to talk about proteins, which we're about to talk about, and nucleic acids. And I would say proteins and nucleic acids, as far as this semester is concerned, they're tied for importance. Because we're going to talk about proteins throughout most of the semester. A lot of the stuff we talk about involves proteins and nucleic acids. Of course, when we talk about things like photosynthesis and respiration, we'll also be talking about carbohydrates. But again, in my opinion, this is probably more important as far as understanding stuff that I'm going to teach you later in the semester. So, if you're not paying attention previously, now's the time to do it. So proteins, you're probably very familiar with proteins already. You're going to learn a little bit more about them. They're very diverse. and you'll know what that means later. We're gonna talk about that, how they're diverse. They are polymers, and their monomers are amino acids. You need to know that for the exam. Like I already said previously, a lot of the questions are gonna be based on that, right? I'm gonna tell you the monomer and the polymer, and based on that, you need to tell me which category of biomolecules we're talking about. So yes, the monomer of proteins are amino acids. If you were to take most cells and eliminate all of the water, more than 50% of what's left would be, by weight anyway, would be proteins. So that just goes to show you how important they are, right? So, all of our cells have nucleic acids, and carbohydrates, and lipids, right? All these things that we're talking about, they all, our cells have all of them. That's why we're talking about them. But proteins make up the large majority of our cells, as far as those 4 molecules that we're talking about, right? So that should tell you how important they are. They're instrumental on almost everything cells do. This is why I think it's so important. So later in the semester, when we talk about genes and gene expression and how that works, it's all because of proteins. Going back to the question that Carla was asking, there's different types of proteins, one of which are enzymes. So every chemical reaction that's happening in your body is happening because of a certain type of protein. And without that, we'd be dead. We can't live without those chemical reactions that are happening. We are alive because of the chemical reactions happening in our cells. And they wouldn't happen without those proteins. Anyway, any questions so far about proteins? All right. I should put that up there. By the way, I just want you guys to know, I don't make these memes. I just find them. I'm like, oh, that'll go good in my presentation. My memes are much better. So you guys know, they're actually usually not biology related. Usually pretty inappropriate, and I couldn't share them here. Anyway, like I said, they're very diverse. They have different jobs. You don't necessarily need to memorize this. This is from the old textbook. But you have like structural proteins, like the proteins that's making up our hair, and it's the proteins that are making up that horn. You have storage proteins, right? You'll find those a lot in seeds and eggs because you need to store energy, for example. Contractile proteins, I think when people think of proteins, that's usually what they think of is muscle, right? So a lot of times people think meat, right? Meat is muscle. That's proteins. Transport proteins, we'll actually talk about this a little bit later in the semester. So as far as the semester is concerned, this one is more important than these. Yes, this one is more important than these. These we don't even talk about that much in this semester, but we will talk a little bit about transport proteins. And the very important ones, as far as the semester is concerned, are enzymes. Again, enzymes are the things that make chemical reactions happen. So they're all equally important, but as far as the semester is concerned, those 2 on the right are more important because to understand some of the stuff I'm going to teach you later in the semester, you need to understand about transport proteins and more importantly, enzymes. Let's make it a little bit more complicated. Here's some more information you need to know. Proteins come from a common set of 20 amino acids. So like I said previously in the slide before this, the monomers of proteins, they're called amino acids, and you need to know that. And now to make things a little bit more complicated, I'm telling you there's 20 of them, and you need to know that number too. There are 20 amino acids. This is a good independent work topic too. So you can look that up, like I've said 1.5 million times. What you can do for any independent work paper is say, all right, the textbook says this, and I looked it up. This source says this, and then you can compare and contrast. This is definitely a good one for that. Some sources claim that there's 21 amino acids. You can dive into it if you want. I'm not going to confuse you with that, but if you want to for independent work, you can look into that. Also, these are just the amino acids that life uses, right? So every cell on Earth, whether it be a bacteria or a brain cell in a whale, whatever it may be, they all use these 20 amino acids. But there are actually other amino acids that just aren't used for life. And again, you can look that up. But yeah, think about that too. That's, in my opinion, that's interesting, right? So everything that's alive has proteins, and those proteins are made up of the same 20 amino acids. So another thing you need to know about amino acids is every amino acid consists of a central carbon, and it has 4 covalent partners. So you've got the amino group, the hydrogen, the carboxyl group, and the side chain. You don't necessarily need to memorize that. You do need to know that there is a central carbon. I might ask you about that. And this is the important part as far as exams are concerned. The side chain. Every amino acid has this basic structure. But what defines, again, there's 20 different amino acids. What makes them different from each other, what makes them those 20 different amino acids is this side chain right here. They all have a different side chain. So you could say that, I guess you could say that defines the amino acid, right? So it's what's on the side chain that makes this amino acid different than that amino acid. Sort of like a throwback really quickly, like I said earlier, last week. Anyway, in this chapter, I was saying when it comes to atoms, it's the number of protons, right, that defines the element, right? Because if you change the number of protons, you change the element. Here, I'm telling you, if you change the side chain, you change the amino acid. It's the amino acid that defines, it's the side chain that defines the amino acid. So everything I just said, here it is in writing. Again, there's 3 groups common to all amino acids: the carboxyl group, the amino group, the hydrogen atom. It's the side chain, sometimes it's called an R group, that defines it. Don't worry about this nomenclature. You don't need to memorize that. I'm not going to try to, there's not going to be a question where I ask you, hey, what 3 groups are common to all amino acids? You don't need to memorize carboxyl group or amino group. But if I were to give you a question where I said, hey, this particular molecule has a carboxyl group and an amino group and a hydrogen atom and a side chain, what kind of atom is it? Or what kind of molecule is it? And then based on that information I just gave you, you should recognize it as an amino acid. Or protein, right?
I was gonna say protein.
Yeah, if you put a bunch of amino acids together, you get a protein. But again, 100 level class, so I'm not going to try to trick you like that. If I have a question like this, you're not going to have protein and amino acids as choices, right? It's going to be one or the other. I don't want to trick you. So don't worry about that. So here's a visualization, another visualization. I already showed you a picture earlier, but here's another picture just drawn differently. Again, you got your central carbon, your amino group, your hydrogen, your carboxyl group, and again, the side chain, the R group that defines it. Again, each amino acid has a unique side chain. That gives it its identity. Also, it dictates how it acts chemically. And again, I know we're not chemists here, but look at the structure, look at the shape. You can tell, like, molecules literally do fit together like puzzle pieces, especially proteins. We'll get into that later. So you can just imagine, just looking at the shape of this and looking at the shape of this, those things are going to fit together differently, right? Those are going to fit in different molecules. They have a different shape. And more so than any other molecule that we talked about, because I keep saying this, but I'm saying it again, and I'm really emphasizing it now. It's all about the shape. The shape dictates the function for everything, but especially proteins. So imagine, again, there's 20 different amino acids. They all have a different side chain, and that's what kind of dictates basically their function. And just imagine, like I said, imagine these things trying to fit together. Not only did they have a different shape, which is easier to visualize, but in this particular example, this one is hydrophobic. Not that you need to know that. I'm not going to ask you questions about these 2 things. This is just examples. This one's hydrophilic. So imagine if there was a protein that had those 2 amino acids, and proteins do fold up on themselves. We'll get to that later too. But imagine a protein trying to fold up on itself. These 2 things would repel each other. So the fact that there's a hydrophobic chain and a hydrophilic chain on that protein is very important, because when that thing folds up on itself like it has to, that part is going to repel itself, right? Those parts would repel. Therefore, that's going to dictate the shape, and the shape is so important when it comes to proteins. Again, everything is important when it comes to shape, but very, very much so when it comes to proteins. Anyway, again, I'm going to put a big X to this as an example, or excuse me, as a reminder. Those are just examples. I'm not going to ask you about leucine or serine, just examples. So like I said, the shape is very important. So let's complicate things a little bit more. First of all, does anybody know, I don't know if you can tell, does anybody know what that's a picture of? It's kind of hard to tell.
It's a basket.
Doesn't look like a basket, unfortunately. Yeah, it's not a basket, but I can see that. I need to get a different picture. So it's supposed to be like a crocheted blanket.
Oh, sure.
And I'll get to that in a second too. That is important. Anyway, that's the next word for attendance. Blanket. Blanket is the next word for attendance. And again, as a reminder, if you guys are here, you don't have to send me the words, but you can to get extra credit. And for those of you who are online or watching this video, you have to send the words to get the credit. Also, a good time to remind you, if you came in late, make sure you email me to let me know you came in late, otherwise I won't know. And also, try not to be late. Anyway, when we bring these amino acids together, I know I've been telling you this whole time that whenever we bring things together, that's called a dehydration reaction. That is still true, but now for some reason biologists get really specific and they talk about this particular type of chemical reaction. And so the things that are holding amino acids together, they're called peptide bonds. You do not need to know that. So they are covalent bonds, but this particular type, they just call them peptide bonds. There's nothing different about those bonds than any other bonds that you've learned about. It just so happens that those are the ones between amino acids. The only reason I even bring it up, well, two reasons. One, it's in your textbook. And two, you need to know, again, the monomer is amino acid. The polymer is called polypeptides. You need to know that. So when we're talking about proteins, the monomers are called amino acids and the polymers are called polypeptides. And that's why I brought up this idea, or this term, peptide bond. That's how they got their name. If it were up to me, I would call them polyamino acids. It'd be a lot easier. But somebody somewhere wanted to make things complicated. So here's something that's a little bit complicated, but important that you understand the difference. So I'm telling you, a bunch of amino acids put together, it's called polypeptides. But that's not technically a protein yet, because it's not a protein until it folds up into its final shape. And that's kind of what I was getting at with this. So you already know it's a crocheted blanket, right? But couldn't I also say that that's yarn? If I were to say, hey, what is that? You could just say, oh, that's a string of yarn. And you would be correct. But once it's in that shape, you could also say it's a blanket, right? I'll do another example like I do every semester. Hold on. Oh, excuse me. I didn't do that right. There we go. Hey, what is this? Piece of paper, right? All right. Now, this, that. I used to fix jets, I didn't used to make them. There we go. Now, what's that? Paper airplane, right? But couldn't you say it's also a piece of paper? Right. At first, it was just a piece of paper. Now, it's still a piece of paper, but we can call that a paper airplane because it's been folded into its correct shape. That's the relationship between a polypeptide and a protein. A polypeptide is just a bunch of amino acids put together. It's not considered a protein until it forms its shape. And its shape is very important, just like this. Hey, it kind of flew. Anyway, it was able to do that because I folded it into that shape. If I were just throw a piece of paper like that, it would do even worse than that thing did, right? Because it needs that particular shape. This is how proteins are. It's so important. Their shape is so important because their shape dictates their function. Anyway, any questions about that? All right. Again, here's just a visualization. This is from the old textbook. When you bring two amino acids together, again, in a dehydration reaction, you form a peptide bond, and now you've got a string of amino acids. And again, I don't know why every textbook I've ever read really focuses on this peptide bond, but it's just a covalent bond. It's no different than any other covalent bond. It just happens to be between amino acids. So I don't know why all the textbooks focus on that. But there you go. It's technically a peptide bond. As you might gather by the way I'm talking about it, I'm not going to ask you about peptide bonds in the exam. It's a covalent bond, is what it is. Anyway, any questions about that? All right. Another thing to mention, and this is gonna come back later in the semester, this is definitely worth thinking about. You have thousands of different proteins. I don't mean like, when I say there's thousands of proteins in your cells, I don't mean like, if you were to count them all up, oh, there's thousands of them. No, I mean like there's thousands of different types. This protein versus that protein versus like thousands of different types. Because I mean, there's probably trillions if you were to count them all up, but different types, there's thousands of them. But again, there's only 20 amino acids. So how do we get thousands of specifically very different proteins that have very different functions? How do we do that with only 20 amino acids? It's how we put them together. Just like the English letter, or the English language, right? So we only have 26 letters in our alphabet, yet think about all the words we have. We have over 1 million words in the English language because we just put these letters together in different combinations. Same with amino acids. Put them together in different combinations, and you'll get different proteins. So back a long time ago when I first made this PowerPoint, I haven't updated this part of it. I always update my PowerPoints, but I haven't updated this one. I think that was like in 2016. That's how many words we had. Obviously, I'm not going to ask you that question. This is not an English class. But anyway, yeah. So all these different proteins based on only 20 different amino acids, and it's all about how we put them together. Anyway, yeah. As far as the exam is concerned, this is a very unimportant slide. As far as the semester is concerned, that is a very important slide. Because later on, we're going to talk about this. Because later on in the semester, you can learn how proteins are made. And this is going to be a very important concept at that point. But for now, just an introduction. If you download the PowerPoint, you can watch this little animation that talks about the primary protein structure. Again, can't emphasize this enough. It's all about the shape. For everything, for all the molecules, it's all about the shape, but more so with proteins. And it's the amino acid sequence that determines that three-dimensional shape. Like I was talking about earlier, that we saw those two different amino acids. One had a hydrophobic side chain, one had a hydrophilic side chain. Those two chains would repel each other, which would cause it to bend. These particular these two amino acids, just as an example, have a sulfur side chain, and they get covalently bonded. So because of that, and this is just an example, you don't need to memorize this, but because of that, this protein now has that curve to it, right? And that's just an example. The point is, again, it's the it's the amino acid sequence that determines the shape. Because if this wasn't, if it wasn't those two amino acids right there, then it wouldn't be bonded right there, and that protein wouldn't have that curve in it like it does. And again, the shape is so important. So again, each single amino acid is so important because if you change one amino acid, you could change the structure, in which case you change the function. And we'll talk about that later too, when we talk about mutations, things like that. So anyway, any questions about that? I'll leave you with this. It's called a primary structure, secondary structure, tertiary structure, and quaternary structure. Probably not going to ask you the difference between the four, but it's there. It's in the textbook. Read about it if you want. And the last word for attendance will be structure. And again, if you're online, send those words before 9 a.m. and also send me one sentence or one question based on the information I give you today. That's it. See you guys Wednesday.
I have one question about the lab we talked about earlier.
Yes. And don't forget, if you haven't done the pre-lab, do it now, because when I leave here, I'm going to go grade those. As long as you have it done by the time I get it graded, then you're okay. That was good. Yeah, perfect. Yeah, don't do that right now.
Okay, look at that, because I'll be wanting to do that.
Yeah, we'll do that now. Glad you asked. You have a pen? This is new. They used to ask for grades. It's a lot easier. Correct. Anytime you want.
Thank you.
Yeah, and keep in mind too, like I was saying earlier, if you want more than 50 minutes, we can meet online and you can take as long as you want.
Gotcha. Thank you.
Yeah, class on Friday.
Yes. It's Friday.
Seven.
Huh?
Friday the seventh.
So like last Friday.
Oh, did we? Yeah. We did.
It's. Move on. Like, I was in class on Friday, so I don't know like what I missed or anything like that.
It's post,
It's on the lectures, but I can't find it on the lecture page.
maybe I haven't posted it yet. I'll look, yeah. Thank you. Thanks for reminding me.